Hello and welcome to this Introduction to Microbiology video. I'm Dr. Ali Roberts, and in this video we're going to look at the bacterial flagella, which is a key structure related to bacterial movement. But before we dive in, let's think about how humans move for a second. We use our legs to move from one place to another, often in response to some form of environmental stimuli. Bacteria, on the other hand, they don't have legs. Instead, they rely on specialized structures called a flagellum, or flagella if there are multiple of them. Essentially, they are tiny whip or tail-like nanostructures that extend outwards from the cell and rotate like tiny propellers, allowing bacteria to swim through their environment. Now, bacteria can have a single flagellum or multiple flagella, and their arrangement has specific names. For example, a bacterium with a single flagellum is described as monotrichous, and a well-known example is Vibrio cholerae, the causative agent of cholera. Other bacterium have multiple flagella clustered in one area of the cell, usually at one of the poles, and they are referred to as being lophotrichous. Now, a well-known example of this is Helicobacter pylori, which is commonly associated with stomach ulcers and stomach cancer, as it's able to grow and survive within the human stomach. Bacterium with two flagella, each at opposing ends of the bacterial cell, are said to be amphitricious, and a well-known example of this is Campylobacter jejuni, one of the most common causes of diarrheal diseases within the Western world. And finally, we have peritrichous bacterium, which is where there is no specific arrangement and flagella project outwards in all directions. And I guess a well-known example of peritrichous bacterium is E. coli, which is found throughout our gut. However, some strains are capable of causing serious food poisoning. And I guess despite its microscopic size compared to the cell, the flagellum is a marvel of biological engineering. It's composed of several distinct parts which under optimal conditions allow the flagella to rotate at speeds of up to 100,000 RPM, which put into context, a jet engine of a Boeing 737 spins at around 15,000 RPM, so almost 7 times slower than that of the bacterial flagella. And so we're now going to shift our focus to its intricate structure. Now, as you might expect, since the flagellum is an external appendage, it must be securely anchored to the cell envelope. While there are some variations in flagella structure across different bacterial species, we're going to focus on Escherichia coli as our model organism. And as a gram-negative bacterium, E. coli has both an inner and outer cell membrane with a thin layer of peptidoglycan sandwiched within the periplasm. Now, these layers help to create a barrier between the internal and external environments, ensuring the flagellum remains firmly attached and functional. Now, to appreciate the complexity of the flagellum, we can break it down into 4 major structures. The first is the motor, which is located at the base of the flagellum and generates the rotation needed for movement. Surrounding the motor are the stators, which help channel ions through the membrane in order to power the rotation of the motor. Next is the hook, which connects the motor and acts as a flexible joint, helping to transmit rotational energy to our final structure of the flagellum, which is the filament. And this is the long whip-like structure that extends outwards into the environment. And so now that we've outlined the major structures of the flagellum, let's dive a little deeper into each one to uncover how they work together in order to generate rotation and movement, starting with the motor. So our first structure is the C-ring, which is located on the cytoplasmic side of the flagella motor complex. It's composed of 3 key proteins, FLIG, M and N, which interact with one another, forming circular discs of proteins that engage with the stator, allowing the generation of torque. Now, the 26 copies of FLIG form the upper part of the C-ring and are involved in motor rotation. The middle section of the C-ring is composed of around 34 copies of FLIM, which are responsible for direction switching, controlling the clockwise and counterclockwise rotation of the motor. Finally, the base of the C-ring is made of over 100 copies of the FLIN protein, which is also involved in direction switching, but also contributes to the overall construction of the flagellum. Now, the C ring is closely associated with the MS ring, which is embedded within the inner cell membrane. Now, this ring is composed of around 26 copies of the protein FLIF, which assembles into a circular disc that anchors the flagellum to the membrane and associates with FLIG proteins of the C ring. This allows rotation to be transmitted from the C ring to our next component, which is the proximal rod. Now, this connects the MS ring to other structural elements of the flagellum, as we will see in a minute, and it is made up of 4 key proteins: FLGB, C, F, and G. Now, around 6 copies of both FLGB and C form the basal segment of the proximal rod, associating with the MS ring, helping to transmit torque. 6 copies of FLGF build on this to form the central section of the rod, ensuring the flagellum can withstand the rotational forces generated during rotation. And finally, around 26 copies of FLGG form the distal end of the rod and connect with the hook element. Now, together, these proteins play a key role in transmitting torque generation to the hook and filament, enabling the rotation necessary for bacterial movement. Now, the proximal rod goes through the P ring, which is located within the peptidoglycan layer. This ring is composed of around 26 copies of the protein FLGI, which helps to provide mechanical stability, reduce friction, and anchor the proximal rod within the peptidoglycan layer of the bacterial cell wall, whilst it rotates at high speed. And so we now come to the final structure of the motor complex, and that's the L ring, which is only seen in the outer membrane of gram-negative bacteria. It's composed of the protein FLGH, and around 26 copies of this protein assemble into a ring-like structure that interacts with the lipopolysaccharide layer of the outer membrane, hence its name. Now, the L ring acts in a similar way to the P ring, helping to provide mechanical stability, reduce friction, and anchor the proximal rod within the outer cell membrane of the bacterial cell wall whilst it rotates at high speed. And so with that, we come to the end of the motor section, which generates rotational torque for the bacterial flagellum. Now, attached to the motor complex is the hook structure, which acts as a critical connector between the motor and the filament. Now, the hook is a tube-like structure made from around 120 to 130 copies of the FLGE protein, so quite a few more copies than we've seen for other parts of the flagella. And the subunits are arranged into a hollow helical structure that gives the hook some flexibility and mechanical strength, whilst allowing it to withstand various forces generated during high-speed rotation. Now, in addition to the hook protein, there are some what we might call hook-associated protein, or HAPs, and they are FLGK, often called HAP1, and FLGL, also called HAP3. And these proteins form the junction between the hook and the filament, ensuring structural integrity of the hook. Now, the main function of the hook is to act as a flexible joint, transferring that rotation of the motor to the filament whilst allowing the filament to bend and adjust its orientation for efficient propulsion. Okay, so when we think of the flagellum, we naturally picture the filament as it's the most visible part of the bacterial flagellum. This is because despite being only 10 to 20 nanometers in diameter, the filament can reach lengths of around 10 to 20 micrometers, so 5 to 10 times the length of the bacterial cell itself. Now, the filament is composed of 2 key proteins, FLIC and D, each playing a very distinct role. And for starters, there are tens of thousands of copies of FLIC, also known as flagellin, which assemble into a hollow helical tube that forms the filament's main structure. In contrast, FLID only has 5 copies present, forming a sort of cap structure at the tip of the filament. Now, this cap structure provides a crucial platform for the organised polymerization of FLIC subunits, enabling the filament to grow and repair itself efficiently. Together, these two proteins create a flexible, whip-like structure that acts as a propeller, rotating in a corkscrew motion to generate thrust in sync with the motor complex. And this only leaves one part of the flagella left, the stator complex. This is a critical component of the flagellum which powers its rotation. It's embedded within the inner cell membrane and is made up of two key proteins, MOTA and B. Now, although it doesn't look like it from the diagram here, these proteins form a ring of around 8 to 10 stator units around the base of the flagellum, and each subunit consists of 4 MOTAs and 2 MOTB proteins. Now, MOTB anchors the stator to the peptidoglycan layer of the cell wall, keeping the stator in a fixed position relative to the rotating motor. Meanwhile, MOTA, which spans the cytoplasmic membrane, acts as an ion channel. So how does this generate rotation? Well, ions, typically protons or sodium ions, flow through the channel formed by MOTA and MOTB. They generate a force known as the proton motive force, and this force drives conformational changes in MOTA, creating mechanical torque that is transferred to the rotor. And I guess it's this interaction between the stator and rotor that enables the flagellum to spin at incredible speeds, as we mentioned earlier, up to 100,000 RPM. And this is what it looks like as the flagella rotates both clockwise and counterclockwise. And so, in essence, the stator complex converts ion flow into mechanical energy, acting as a powerhouse of the flagellar motor and enabling the bacterium to swim through its environment with remarkable efficiency. Now, the reason why bacterial flagella are important is because it allows them to move in response to an external stimulus, and we call this taxis. Now, this movement or taxis can be both positive or negative. And so a positive taxis means the organism will move towards the stimuli, whereas a negative taxis means the organism will move away from it. So as an example, a bacterium might want to move towards nutrients and away from something such as an antibiotic. Now, there are many different types of stimuli that affect our bacterial cells. So bacterial cells can respond to light, and we call this phototaxis. They can respond to water, and we call this hydrotaxis. They can respond to oxygen concentrations, and this is known as aerotaxis. Now, one of the more common taxis out there is where cells respond to chemicals, and we call this chemotaxis, and we'll have a look at this in a different video. Now, there are some other less known taxis. Cells can respond to electric fields, known as galvanotaxis, magnetic fields, known as magnetotaxis, and ionic strength, known as osmotaxis. Now, for each of these taxis, there are two sides to the stimulus. We can have attractants and repellents. And attractants are favourable environmental signals that bacteria move toward, while repellents are harmful or stress signals that they move away from. These signals are detected by receptors which activate a signalling pathway that controls flagellar movement towards the attractant or away from the repellent, allowing bacteria to navigate their environment for survival. And with that, we come to the end of this video. Hopefully you found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.